so far for such a young man. Alessio did his degree in Florence. He then did his PhD at Cambridge with the late Professor Chris Abel at the forefront of the field. Before you came up to Bonnie Dundee, what a good decision. In 2013, and by 2016, you had a personal chair in chemical structural biology, and you are the founder and director of the Centre for Targeted Protein Degradation, TPDs, which we'll hear about more tonight. I have to say, looking at your CV, Alessio, you've done you know, lots of awards and very much at the forefront of his field. I have to confess, I watched a little YouTube video this afternoon of Alessio, so I have know a little bit what he's talking about. But that was for the Cambridge Philosophical Society. That shows you the kind of research that he's doing. I think we might hear the word ubiquitination tonight. Will we? Yeah, ubiquitin. Some of you may or may not know. It certainly wasn't in my undergraduate books. It's new-ish. So anyway, it is truly cutting-edge science, and I'd like to welcome you, Alessio, onto the stage and give us your talk. Thank you very much.
Thank you very much, Rob. Can you hear me from here? Because I'm still recovering from my voice going completely down last week. And I just got back from Boston, where I was giving a talk and sitting on a panel. And yes, I sort of recovered in time for my voice to speak. And it is good that he's still here. I must say, I'm really excited to be here. I'm also slightly nervous because these are always the toughest talks. In particularly, I'm nervous because I've heard a lot about Curious Minds. I've heard a lot about this series. In fact, one of my very talented postdoc lives in Perth, and she tells me, oh, I often go there, and I did Cafe Science last year. And you know, she actually is involved in suggesting names, and you know, is often going there. I really enjoyed that as well, and I hope that in the next sort of hour or so, and hopefully we're going to have a lot of discussions as well afterwards. That you know, you'll come back. Getting a bit of a flavor of why these things that we're doing is so exciting, why it's so cutting edge, as Rob was saying, and also understanding a bit more the principles of the science behind it, and how, because it is so innovative and we're still at the cutting edge, how innovation still is an incredible part of it, but also how you know, inspiring people and collaborating with people is so critical. So the Center for Targeted Protein Degradation that we just created, it opened in January last year, of which I'm very fortunate to be the director. We have these 3 core values: innovate, collaborate, and inspire. And I hope you'll, you know, throughout the talk, you'll see a little bit of that. So my name is Alessio Ciulli. As Rob said, I've been at Dundee since 2013, and I've never looked back ever since. It's a fantastic place to live. I love Scotland too, and I'm, you know, I've got, this is my email address. I'm also quite active in social media these days. Is quite a thing to do, and I really enjoyed it. It's a great way to connect and to get immediate feedback on your work. We post all our papers immediately online. Make it available, and that is another thing of ethos that, you know, we do, and is part of our Inspire. But I just wanted to say that please do get in touch, you know, with me after this lecture if you have any questions, if you want to learn more. These are my disclosures. They're not very relevant to my talk, but just to say that, you know, I have some interests because of the company that I co-founded, that I have some shares on, and also because many pharmaceutical companies have been and continue to fund research on our lab. But I just want to start to say, for me, a passion is really important in what we do, in everything we do in life, right? And as scientists, I think science is really one of these things that if you have to have a passion for, you have to have a sense of mission, but also you have to really love what you do. And I trained as a chemist, so I studied chemistry. So I'm a chemist by background. And actually coming to Dundee has been wonderful because Dundee is so strong in biology. And so I've been able to absorb quite a lot of that. And I'm still trying to get my head around how biology works. But I've learned one thing, that biology rules. So I'm quite humble as a chemist to say, yes, biology rules, but as a chemist, we can manipulate it. And so I hope I'm going to show you that we have found a new way to manipulate biology. And this new way to manipulate biology is really important because it's a new way to make medicines, right? It's a fundamental new way to develop drugs. But yeah, this was the kind of thing that got me hooked when I was a kid, okay? I have no idea of essentially 99% of what I'm going to tell you about. There's going to be very little chemistry in my lecture, in that I'm not going to go into the detail, for example, of how we make the molecules that we made. But I got excited about things like chemical structures, chemical formula, and reactions. And I just got excited about this idea that with chemistry, we can actually make things. We can make new things, right? Somebody said, to understand something, I have to make it, right? And the other thing is, when you make new things, you suddenly, for the first time, have something that has not been created before, right? That perhaps is not available in nature. And then I learned, these things could actually be used to develop medicine. And I learned that these medicines actually work because they modify something in the cell or of an organism, whether that is a bacteria, virus, or our own human cell, for example, in the case of cancer. So that was my passion. The other thing I really want to say that is important is that it's often underestimated, but as scientists, our journey is very much influenced by the environment we go through, but also the people that we that we encounter throughout our journey. And I was very fortunate in my career to be mentored by really incredibly talented chemists, structural biologists, who taught me a lot. And then I went to the United States for a brief stint after training in Florida, my hometown Florida, and then in Cambridge for many years, as I felt I needed to experience the US is a unique place. But actually, this is a very important, even if very short 4-month stint, a very important seed to a key collaboration for the field with one of the pioneers of the field, Craig Cruz from Yale. Then I started my own independent career, and actually one of the things that I think I started reflecting more and more is how your mentors and the people you train with and that almost act as role models for you, actually how they influence how you think and what you do. And one thing I realized is all of these people were very entrepreneurial. They span out their own companies. And so, perhaps not surprising, I started some entrepreneurship, getting hooked by entrepreneurship early on. But then, perhaps not surprising, right? I have found to be quite, I wouldn't say comfortable, you're never that comfortable, but at least, I had that experience and that reassurance. If they've done it, I can do it. And so I've also founded myself initiatives such as New Spinoff Company and the Center for Targeted Protein Degradation. And so I think these things are important. Science is done by people, and also, sometimes to enable best the science, you have to kind of realize that you have to do something outside of your comfort zone. You have to create something, very much like a chemist creating a molecule that's never been seen before. And there's that level of imagination, that level of creativity. Sometimes, in the same way, you have to think about how to create new things that then enable and open up new areas. And I think these are big creations that I've really enjoyed facilitating. So what am I going to talk about? And I actually started working very early on in biochemistry, or rather chemistry to study biology. And I still remember Ivano Bertini in Florence, the late Bertini, who told me, Friolino, he was big voice, right, and in a very Florentine accent, if you want to be a chemist these days, either you do materials, you're going to have a talk on plastics in 2 weeks' time, right? So there'll be chemistry there for sure. Either new material, or you study, use it to study biology and drug discovery, and today there's a new avenue there, and I got hooked. And they were studying proteins in that lab, big NMR spectrometer, nuclear magnetic resonance, to study how proteins look like, the structures of the protein. And I got hooked into studying how proteins bind small molecules, okay? And that's important. That's important because our human cell contains many, many genes. We now know what they are exactly after the sequencing of the human genome. We know that there have about 10,000, 20,000 in some cases. And these genes code for proteins. And proteins are really the working horse of our cells. They carry out key reactions like enzymes. They act as transporters. They act as key components of the organelles of the cell, and they really mediate all the various aspects of how cells live and die. And so proteins actually have a life inside the cell. They're born, they interact with each other, and ultimately they get, once they're old or they're no longer used or needed for a particular task, the cells get rid of them, so the proteins die, right? And often naturally, as part of the action of the protein binds to small molecules. These could be, small organic molecules, they're often called cofactors or coenzymes or metabolites if they're already part of nature. But we as chemists, we can create small molecules that nature had never seen before. And these small molecules can impact the natural process of the cell. So we can have small molecules that can impact any of these processes. But actually, and importantly, these small molecules can modulate, either revert or trigger aspects of the way the cell works that can ultimately result in curing a disease. So for example, in cancer, what most often happens, almost always happens, is certain types of cells go awry. They become, let's say, unnatural. Certain features of how the cell naturally, physiologically work goes wrong. And that going wrong is typically triggered by some bad proteins or disease-causing proteins that drive that cancer. Typically, the cancer proliferate and start growing, or has some other types of features that are hallmarks of cancer. Many cells are viruses or bacteria, and then we get infections, and we have to think about how we treat them. Many cells are cells of our immune system, right? Whether innate or whether adaptive, these immune cells are critical to how we react to external pathogens, even in cancer, how we mediate. And so how our small molecules might impact those immune cells can ultimately generate medicines that can ultimately cure patients. And so it became very clear to me early on that as a chemist we have an incredible opportunity. And in fact, it was very clear that most drugs worked by binding to proteins, by modulating proteins, maybe blocking some of these enzymes, right? And so from understanding protein to understand why they're important in cells, to us chemists realizing that we have that opportunity, I got hooked into drug design. So the design of this molecule. How do we do that, right? And what are the principles of how we design these molecules? And most importantly, how do we do this rationally? And then I understood very early on that to do that rationally, we needed to understand how these small molecules bind to this protein, how they interact with this protein. So I got hooked by these interactions, protein small molecule interaction, often called protein ligand interactions. Then I learned later early on that many proteins interact with other proteins, or protein-protein interactions. And with small molecule, we can modulate that, but I'll get to that in a moment. And before, I just want to sort of give you an overview of how conventionally drugs have been developed. And there's been waves of this, and so the Curious Mind Society was founded here, and God knows what kind of drugs we were using, but eventually a wave of drugs such as aspirins, and more drugs that we realized, right, that they had some sort of an effect, Chinese medicines, other types of so-called phenotypic drugs in patients were being used, but we didn't really know how they were working. Until then, around these times, scientists started to be able to crystallize proteins, right? Pioneering work from many scientists, including Nobel Prize winner Dorothy Hodgkin and many others, started to crystallize proteins, and that target protein then began to illuminate that actually there was an opportunity to design drugs rationally. A very good example is a drug called Relenza here, which is a neuraminidase inhibitor, is an anti-HIV drug. And these drugs bind to the target protein and block the activity of the target protein. And so these types of inhibitors have emerged ever since and been more and more designed rationally. And this works. And they're called so-called small molecule drugs. So our work is in this space here, and has always been, at least for me. But I just want to highlight for your awareness that there are also drugs that are so-called biologics. They're typically antibodies. And we've become better and better, especially in the last couple of decades, at developing these drugs. And again, they work in the same way. They bind tightly to a target, typically target extracellularly, because the good thing about these small molecules is they're good at getting in the cell. But these antibodies aren't. They're quite big proteins, so they can't get in the cell very easily. So they're very good to target the whole cell or target extracellularly the cell, mediate immune systems and whatnot. You may have heard of immunotherapy. Okay, what's brilliant about this is they're very specific, they're very potent, right? But they're very limited. You can't easily target proteins that are inside the cell. So what has happened in the last few years is this revolution whereby we're kind of developing molecules that have the best of both worlds. They're small molecules, but they have activities like biologics. And they can target protein target inside the cell, like the small molecule. They're not limited to outside the cell or targeting the whole cells. And they induce proximity between proteins. So instead of blocking a single protein target one-to-one, we bring two proteins together, right? And so it's beyond this concept of one-to-one, we now induce a new interaction. And these are protein-protein interactions. And so it's a single drug that now brings the specificity of nature, like a biologics, to a small molecule. And this has really been absolutely revolutionizing. And so for the purpose of this talk, I'm going to focus on small molecules that are called degraders. Okay? And it turns out that the way these molecules work is the second protein that comes and gets recruited into proximity is a protein that is part of the natural sort of garbage recycle pathway of a cell. So the natural process that trims down unwanted or old damaged proteins. And the major process that the cell uses to do this is a pathway called the ubiquitin proteasome system. And so without having to go into the detail of that, of ubiquitin and the proteasome and all the components of that, I'm just going to try to leave it as a bit of a black box for you, although I'm going to show you some representative structure. But I want to hopefully inspire you to say this is important. It got recognized by the Nobel Prize in 2004 to these three scientists, Aaron Chechanova, Aaron Hemschko, and Irwin Rose, for their discovery of what's called ubiquitin-mediated protein degradation. So you'll see a bit more about this. But the bottom line is, there's been a Nobel Prize since 20 years ago, and these people have made important discoveries. So it's almost like our science stands on the shoulder of these giants, right? So I was, that year I was in my second year of my PhD, right? So just to give you an example. I actually didn't really know much about ubiquitin too. But this is probably the first evidenced cartoon type drawing that illustrates the concept and this new idea. Dates back to a patent filed in 1999 that actually was never prosecuted. It was a company called Proteinics. Nobody, took notice. But they hypothesized. This is the beauty of, some ideas and a piece of paper. Maybe you patent them, but it doesn't quite matter. That idea, that vision, that projection of what might be possible. They hypothesized that one might, perhaps with a small molecule, perhaps bifunctional in nature, and they draw this small molecule like this sort of piece of bone here, or dumbbell. Horribly looking, but nonetheless, anyone has imagination about our drawing skills, but might recruit at one end the target protein. So if you think about it, that was the target protein I was showing you earlier, and this bit is almost like that inhibitor, that small molecule that binds. But this is bigger, this is more complex, is bigger such that at the other end has a recognition element, meaning maybe an inhibitor, maybe something that binds a different protein. And that protein is a protein that works as part of the ubiquitination system. They call it just ubiquitination system in this patent here. And by doing that, they would bring the ubiquitination system in proximity. This is how this ubiquitination system works. It fires a small protein called ubiquitin to a protein that naturally needs to be removed. That ubiquitin gets tagged onto that protein, almost like as a signal. It's like you put some post-it, on something as a signal. Please trash it. Send it to the garbage bin. Okay, and they hypothesized that by doing that, potentially we could fool, or rather, say better, co-opt, hijack, if you wish, this ubiquitination system to work on a target protein that we want, rather than on the target protein that this works naturally to. And so if this target protein is a disease-causing protein, then perhaps that might be a way to develop a novel medicine. Now, you can talk more later. This didn't take much notice. One of the things people said is, this is never going to work. It's already hard enough to make a single inhibitor or a single drug for a single protein. You're now telling me that you're going to make a drug that recruits two different proteins? This is too complicated. It's never going to work. But some scientists took notice, and very early on, in fact, since 2001, shortly after, a paper was published that kind of showed some proof of concept of exactly this idea, right? In 2001, and this proof of concept molecule here, this dumbbell, was called proteolysis targeting chimera. Chimera it's like a monster with two heads, right? So this was the monster with two heads that triggers proteolysis, because ubiquitin-dependent proteolysis, ubiquitin-dependent degradation, is ultimately what was going to happen. But this took years ever since to get any traction, in large part because the molecules at the time weren't really optimized. In particular, I'm drawing you here still the target, which is now here, and now this ubiquitination system here, I'm drawing it a bit more complicated. As you can see, there's different bits in here, because by then we knew more. We know that this ubiquitination system is composed of different proteins, E1, E2, E3, And the details doesn't really matter, but they're different proteins that work together, and they're called colin ring ligase. These are the enzymes that fire ubiquitin, right? And so it's putting a bit more nuance to this big picture. And they fire ubiquity coming from this E2 because it's attached here. This E2 is recruited to this different protein from this end. At this other end, you have another protein that normally recruits the target protein that the cell needs to get rid of and ubiquitinate naturally. And with this dumbbell in the middle, where you have a ligand or an inhibitor for this, and a ligand and an inhibitor for this, and a little linker, you can bring this together. And the problem was, we didn't have good ligands for this at all, right? Mm-hmm. And so this was where we were able to innovate and sort of, 24 years down the line now, this was a key research that really, enabled the field, that really provided the tipping point. Because moving from more peptide-like natural ligand of this E3 ligase, we developed, initially in collaboration with Cruz, my lab and his lab, much more drug-like molecules. Molecules that in their own right could be drugs, could be medicine, that look like medicine. And industry started to take notice. And then, a few years later, in my laboratory, we made the first such molecule, where we linked this molecule by a linker, and this is a polyethylene glycol. So we have little chains, linear chains of carbon atoms and oxygen atoms. But it doesn't really matter, it could be any chemical linker, linked to a ligand for a target protein. And then we showed that molecule, that this PROTAC, now composed of this new molecule, worked in cell. It degraded this protein really rapidly in cell. That got us excited as crystallographers. We wanted to see how this looked like. These have never been seen before. And for the first time, we were able to solve the structure, crystallize the protein with the bound PROTAC. But we couldn't just solve the structure of the protein like Dorothy Hodgkin, spent years and years and years of doing in the 50s or 60s, and now you can press a button and solve the structure. We had to first form that complex. And so that's a complex of our E3 ligase, our ubiquitination system, with the PROTAC in the middle and the target protein, simultaneously bound. And now, in a way, you can see that this is a high-resolution image of that, right? Just twisted 180 degrees. You got our target protein, you got a ubiquitination system, you got your dumbbell in the middle. And that's what it looks like. So this was the first ever time in the field that we saw what a molecule like this looks like. And just fast-tracking to say that there are now over 50 of these types of molecules that are being tested in the clinic. I'll tell you a little more about it. Why? Because with this type of molecule, we are now able to remove very rapidly these target proteins from the cell, as opposed to just blocking them. So really is the innovation. And this is often what needs to happen in science. Innovation is not a straight line. It's not like you get a big idea and you draw it on a piece of paper and it happens tomorrow or in a month or in 6 months. It often takes a lot of pivoting, a lot of zigzagging, and a lot of key innovation happening that all influence the next steps. And it often takes a long time. So just to summarize for you, this is how conventional drugs bind. This is how those inhibitors I was telling you bind. They block the target protein, and what that requires is that we saturate our target protein. If our cells have 100 copies, 100 molecules of that target protein, they usually have many more, thousands and millions per cell. It doesn't matter. Let's say 100. Then we often need to block 99 of them, or 99.9%. You never quite block all 100. And that's what I trained on, with Chris Abel and Tom Blundell, pioneers of ways to do this. Exactly this, or how to design those molecules. But we always kept, working in that space. And it doesn't really matter. What I'm showing you here is that this was cool, but it was really hard for many targets. There were lots of limitations, and I wanted to explore new ways of applying what I had learned to new ideas. Okay, and so then this new concept of potentially attempting to degrade the target really hooked me. Why? Because conceptually is extremely exciting and different. It basically means that we could have a molecule that works like an enzyme. So enzymes are catalytic, meaning that they go through multiple cycles. So a single molecule of an enzyme can catalyze 100, 1,000 reactions over a certain period of time. And what if our molecules could do the same? An inhibitor cannot. An inhibitor binds one-to-one. So to bind 100 molecules per you need 100 molecules of inhibitors. And so here's the idea. If it's degrader, I would call it degrader, could just bind transiently to the target. Then we remove the target. But the target is degraded, but the degrader is not. The degrader can go on to the next target, and then to the next target, and very much act like an enzyme. So catalytic, meaning it could act at low concentration. Maybe we only engage 5 molecules of our target at any one time. And over a period of time, maybe hours, but if we got very good degraders, sometimes minutes, we can degrade all of those 100 molecules. Right? Also, the drug could bind anywhere on the protein. Because remember, the dumbbell only have to engage on the target. It doesn't have to block a functional site. So we could hook to that target, grab it anywhere, bring it close to the mouth of the dragon, the ubiquitination machine, that then flag it. And nature doesn't care if you're grabbing it from a functional site or not. You'll just see it and it will start firing. And this is another really important aspect. Now the target protein is completely depleted from the cell. So it turns out, I'm sure you heard of CRISPR, right? The new Nobel Prize winning method to do genome editing. That's a great way to knock out a gene. What you do there is you tamper with the DNA so that cell of that animal no longer expresses that protein. So it's a knockout for that protein. That protein is not there. The industry, the drug hunters, and many people use that as a proxy to say, this is a good target for disease X. I've made the knockout, the cancer no longer grows. And then typically in the past, we use that information. Prior to CRISPR, we would do RNA interference. It doesn't really matter. We use that information to say, let's make an inhibitor of it. But actually, if you think about it, when you inhibit, your target's still around, and you're blocking a single interaction of that target, a single site, ideally the functional site. That's very different from removing the protein altogether completely. Whereas these degraders would do exactly that. So we now know that what's really critical is the mechanism of action, going by the ternary complex, and also that structure was really illuminating. So here is now a bit more of a higher, a sort of more detailed picture of what happens after you do that proximity, because this effective protein carries that ubiquitin, which is the hot potato or your dart, and it's firing it to the target to try and hit the bullseye, right? And then once the target is tagged, it gets degraded very rapidly. The cell takes care of it. So we as a chemist, all we have to do, and it's not easy, we have to find a ways to do exactly that. And at the time, we didn't really know how to do it. We didn't know what a good degrader looked like. We had this dumbbell, 2 ligands, 2 inhibitors joined by a linker. And all the drug hunters, all the industry med chemists would look at it and say, this will never be drug. So, in the early days it was really tough, right, to get any serious, to be taken any seriously in this space. And so, this is just to tell you that this wasn't just a pattern. It turns out that nature does this mechanism a lot. So nature is always an incredible source of validation for ideas. It turns out that viruses have been evolved to do this very well. They hijack the host cell, And then they express proteins that work exactly like degraders. So the viral protein, in this example, is this protein called VIF, hijacks the natural host ubiquitination system to bring into proximity a host cell antiviral protein. In this case, it's called this ApoBac protein. Is an antiviral defense protein, so that it gets degraded very rapidly, so that the virus is no longer having to deal with this inconvenient host antiviral protein and can then reproduce and go on and infect the next cell. It turns out that plants actually also exploit exactly the same mechanism. And I think there's a lot that we can learn from viruses, bacteria, and plants, right? And this becomes over and over again. CRISPR was developed, upon key fundamental discoveries between, how viruses, infect bacteria and whatnot. And so this is a natural mechanism for plants that utilizes a small molecule called auxin. Is a plant hormone. It's a metabolite. As a growth hormone molecule that triggers plant growth and development. It's a very small molecule. It's like tryptophan. And the way auxin works is when you have a lot of it, it acts as a degrader. It acts as a glue. It brings together to the ubiquitination system of plants, in this case, a natural transcriptional repressor protein, here drawn as in red, which then gets degraded, and because it's a repressor, you remove the repressor, you remove the brakes to transcription and growth. So a lot to learn, right, from nature. But again, those are natural molecules. And here is just a bit of a more picture of this E1, E2, E3 mechanism. That's your ubiquitin, the hot potato that binds at that right end. And now here is where we recruit with the dumbbell target protein. We call it substrate or neosubstrate. And that's how you fire the ubiquitin. You build that, you tag that protein. Once the protein is tagged, it gets recognized very quickly, unfolded, and very rapidly degraded by the garbage bin of a cell, the proteasome. So as I said, this happens once we put the chain here. So what we really care is to develop things that do that bridging there. And that's exactly where our degrader sits and where it comes from. This is just to show you that 2001 study, the dumbbell and that paper, but these molecules suffered because they were very large. They had peptidic moieties here. They couldn't get inside the cell. They didn't have the properties to become a drug. So together with Craig Cruz, we realized this is where we need the attention. And we need to move away from the peptide, and we need to make a more drug-like molecule. That's what we did. And that's where we were able to leverage my own training in fragment-based drug design, because this is to say a little bit more about column ring ligases. It doesn't really matter now. And this is just to say that we focus on a particular ligase. So is this protein called von Hippel-Lindau. Without going too much into the detail of the information, I just want you to know it's a very important protein, and it works in a very important process. And because of it's so important, it's ubiquitously expressed. It's everywhere in all cells. It works in the process of sensing oxygen. So we are aerobic species, right? We live thanks to oxygen. We need oxygen. And we have evolved a very cool way to respond to low oxygen levels called hypoxia. So basically, we have a system that gets activated like a sentinel when oxygen level go down. And to do that, when the oxygen level is normal, oxygen basically acts as a signal to mark for destruction a key transcription factor that responds to hypoxia, to low oxygen level. And VHL is the enzyme that catalyzes a very rapid ubiquitination of that HIF under high oxygen level. When oxygen level is low, this protein escapes that recognition, no longer gets ubiquitinated, and then goes on and trigger a response to hypoxia. So I realized this is quite complex. It doesn't really matter. What I just want you to realize is we thought, hey, hang on, this system must have evolved to be extremely efficient. What if we bring our disease target protein, our disease-causing protein, in the mouth of the dragon there instead of the natural substrate? We're going to get good ubiquitination. So to do that, we realized that we had to develop more drug-like small molecule, right, as I just said. And we leveraged some crystal structure that had been previously solved. And look how beautiful this is. This is a natural peptide epitope of that transcription factor that binds to VHL. And it binds to it thanks to that key modification of that. It's a proline residue here. And that modification is hydroxylation. It becomes a hydroxyproline. Hydroxyproline very famously identified in collagen, very important structural protein. But this is a signaling modification, and that's a hydroxyproline that binds. I couldn't be hooked more. You know, I told you I studied on protein ligand interaction. This is very specific. I saw this and I said, you know, Craig, we could use this as a hook. Nature has given us a starting point, has given us that fragment that we can start elaborating. So we got excited, started putting together a team, and together, over many, many, many years, since that visit at Yale when we started working on this and started theorizing this, from the two laboratories with key contributions from chemists and structural biologists in Craig's and my lab respectively, and then later more and more work from my lab and others optimizing this, we went from that hydroxyproline, very, very small, very little, atom by atom, and we built that VHL ligand that then we used in the dumbbell, in the process. And so lots and lots of work over many, many years, lots of key publication. Industry started to take notice. And this is where we started to feel, okay, we're in for something here. GSK, you know, invested in our labs through establishment of a discovery platform unit. Craig, very entrepreneurial, spun out his own company in 2013, and then I spun my own in 2017. But I just want to take you through a journey. This is a journey of the VHL ligand. And this is what was homemade, right, by our laboratory. That's the chemists that design and make that new molecule that is going to transform the field. And that's what it looks like. You know, and for a long time, we actually weren't sure if this was going to work, because this starting point is so small, is so weak, it cannot work. So we had to build it. We had to get it bigger and stronger. But that's it. That's the starting point. That's what nature gave us, right? And that's a VHL here. And that's where it binds. And that's the structure my postdoc Inge van Molle solved for the first time, seeing that hydroxyproline bound where it's supposed to bind. Look, this is how it binds in the peptide, okay? And so even if you're not a chemist, you can appreciate it looks the same, right? So that's our hook, that's our fragment. And then we build on the left, we build on the right, and even if you're not a chemist, you appreciate, and green is carbon atom, blue is nitrogen, red is oxygen. It doesn't really matter. We are optimizing. We're fitting this thing on that binding pocket, right? That recognition element, right? And we're making it more specific, and we're making it better. And we're doing it now in a way that even the natural peptide doesn't do. And then we realize, haha, here, look, is mimicking the peptide. If we just mimic the peptide here, boom. So the peptide has guided our design. Nature has guided our design. But we've come up with a molecule that nature has never seen. Nature doesn't know that we have designed it based on a molecule that it has and that it binds there. We are designing now a molecule that is new, that is unnatural, and that we can use to do things differently. So that molecule that you see here is the molecule that is now used in all those types of protein degraders that work with this ligase, okay? And so it turns out, and this is just to tell you, we reviewed the field and we took the journey, we reviewed the journey of how we went from that amino acid to initially from the peptide and natural ligand to the inhibitor to those bifunctional molecules. So I want to take a few minutes to actually tell you that we were not the only one, right, in the field excited about this, but actually something really incredible was discovered coming from a completely different angle. And this is the discovery of how a very infamous drug called thalidomide works. I'm sure you've heard of this. You know, this drug prescribed in the late 50s as anti-sedative, especially for treatment of morning sickness in women during pregnancy, led to phocomelia and the realization that many of the kids that were born as a result of that correlated, you know, with these developmental defects. That's a molecule, very small. It almost looks like a hydroxyproline, but incredibly potent, and it did all of that. Turns out many years later, because very active, a company called Celgene then repurposes initially for leprosy and then for multiple myeloma. And these molecules are often called immunomodulatory drugs because they have a very pleiotropic activity. And in the right context, a molecule similar to this has become a blockbuster drug called enalidomide. But scientists didn't really know how this worked. How does this molecule work? How does it trigger that phocomelia and that developmental defect? And how does it work as a drug, as anti-cancer drug? And we didn't know what the target was. So unlike our approach, we started from the target and we developed the ligand building on the nature hook. Here we had a drug, but we didn't know what the target was until Hirohanda in Japan, and without going into details of how exactly he did, he mobilized the compounds into beads and took cells, broke it down into pieces, but maintained the proteins intact, flow them down and asked the question, what pulls down? What do these molecules bind to? And he found a band, and this band was this protein cereblon, which came down with another component of E3 ligase. So cereblon is an E3 ligase, and it's a target of this. So everybody thought, okay, this works by inhibiting the ligase. And in fact, a crystal structure was later solved by colleagues, structural biologists in Basel and Celgene, that in fact validated and showed this molecule indeed bound, like our crystal structure that showed hydroxyproline bound at the heart of that ligase. They show that thalidomide and its cousin bound at the heart of this cereblon, right? And so how does it work? It still doesn't tell us how it works. And we all thought it was an inhibitor. Instead, in itself, it's one of those dumbbells. It's just small. It's called molecular glue degrader. But what it does is it brings together the ligase to a neoprotein, even by being so small. That was remarkable, right? And it turns out that then it is these proteins that get degraded, and it is the degradation of this protein that actually results in the pharmacological activity of this molecule for treating multiple myeloma, and in this case, particularly for treating another type of hematological malignancy and by a different target. So how remarkable, huh? So around the same time, this is just a slide to say how that little thalidomide analog, how much of a blockbuster drug it has been. And so suddenly, you know, we had showed that we can get a drug-like molecule on VHL. These scientists have finally figured out a bit more about how thalidomide works, and have shown that a known drug approved by the Food and Drug Administration as a medicine used in the clinic to treat patients with multiple myeloma worked exactly by this mechanism. And so that really revolutionized the field, because then, around exactly that same time, from work from us, which I just told you, and that's the same picture you saw earlier, and work from these other folks that I've just told you, reviewed as I just said, and here's another review in this same journal and issue, we now had two of those recognition element ligands with crystal structure of how they bind, right, with very high affinity, and they could be our E3 ligase ligand, that then we can attach a linker to our target protein. And we now know actually that these can also work as degrader in their own right. And so that's, that's a hook, and that's the other hook here. Beautiful, right? It's all in the molecular recognition. Boom, 2015, my laboratory in Dundee already at the time, and then Craig Crew's laboratory and the laboratory of Jenny Bradner in Harvard, and Arvinas. Around the same time, we showed different molecules that all contained either of these two ligands, the VHL and the cereblon, degrading different types of proteins. So this really was now the moment where industry took notice and said, ha, these are going to become drugs. So this is just to tell you a bit more. I love structures and how we were then able to show that this molecule here, how it binds, and this is a structure. And it's a beautiful, realization. This molecule glues these two proteins together. And so we've got our VHL ligand here that we designed and developed. We've got the target protein ligand at this end. So the target and the ligase come together and form now these induced protein interactions. All these, you see interactions with the linker. These interactions are highly specific. We bury a lot of surface area. We're really gluing these two proteins together. So now that's the induced proximity. Our molecules bridge these two proteins together. And back then, we didn't have any of these molecules being treated in patients. It was only 2017. 2019, the first PROTAC drug entered the clinic. As of today, there are over 30 of these types of molecules in the clinic. Many of them are cereblon, for a reason we can discuss. Quite a few of them are VHL. Some are not disclosed. And we just wrote a review on this. That should come in press. Many of these are for treatment in cancer, which are listed here, but there are also others that are starting to emerge for treatment of diseases beyond cancer, notably immunology, but also neurodegenerative diseases, not listed here. It's just lagging behind. And here is some of the chemical structure of the molecules that are being tested in patients, right? And they all contain those recognition elements, those little ligands for the ligases shown here. And it's really been incredible to see how the field has evolved and reached a stage where these molecules can now be more rationally designed. I showed you earlier a crystal structure. We were able to crystallize a ternary complex. It turns out it's often really hard to crystallize proteins, and especially crystallize large beasts, like the big machine of this ubiquitination system. And revolution in cryo-electron microscopy, again, recipient of another important prize, Nobel Prize in Chemistry, our own Richard Henderson from the Laboratory of Molecular Biology here in the UK, one of the pioneers, have really revolutionized the approach so that now we have these electron microscopes. So number one, you don't need to crystallize a protein, and many proteins don't like to crystallize, and so if you can't crystallize a protein, you can't solve the structure, because you need the crystal to do that. All you need to do is generate good samples, and then you can freeze this sample, and you can shine electrons to it. And it's just been incredible what a revolution this has been. We realized at Dundee that we needed to catch up on this, and in 2022 we got our first microscope. And in 2 years, I've just been blown away by the kind of structures that we've been solving. And I just wanted to show you one, particularly because it's been solved and it's been championed by a very talented PhD student of mine, and she just defended her PhD thesis Viva today. Right. So this morning she gave a lecture, and it was absolutely outstanding. And then she passed the Viva with flying colors. But I could only dream when I started working in this field of actually seeing what this looked like. She started in the lab and she modeled this, but then she went on to solve the structure. And this is a real structure. She generated the particles that look like this, and where you can see here how at the mouth of the dragon you're recruiting the new protein, and you can see how the ubiquitin is coming close. You can see the next protein-protein interaction happening at these ends. So we can see where the hot potato, where we're firing on the target protein. And so this has helped us to understand more about this ubiquitin ability, because ultimately you have to tag this protein. So we started to understand in incredible level of detail this process at atomic level. More and more, we're starting to see what needs to happen for these degraders to be good degraders. And this is critical and it's so important. Why? Because it informs how then we go back to the lab to design the little degrader drug-like molecule that is ultimately going to be tested in patients, right? And so this paper just published a few weeks ago, and actually other fantastic groups have also published in incredible papers as well. So we now know a lot more about how these degraders work. They have to go through this cycle like catalysts, and without going into the details, we've dissected every single step in the process. We now know a lot. This ternary complex is really critical, and it really matters whether the degraders can go through this cycle very well, because it's a whole difference between not degrading a protein. I'm showing you here those 100 molecules of target proteins to begin with, like 100%. You can have flat line, you get no degradation, or you can have slow, incomplete degradation, or you can have fast, complete, effective, durable degradation. And arguably, you know, as a drug hunter, you want something that hit like this. Although sometimes you might want something like this, you still want to maybe leave 50% of your protein. It's good to know how to do that. This is just to say we degraded a lot of targets in the lab. 3 of these are disease-causing proteins. And we, amongst many others, have been contributing to the development of molecules like this, including a molecule that degrades one of the key drivers of cancer called KRAS. This was a protein previously thought to be undruggable. We, for the first time, have developed a degrader that can hit and deplete 13 out of 17 of the mutants, the oncogenic mutants of this protein that drive mutations. This is important for things like colorectal, pancreatic, and lung cancer, for which patients typically have nothing. There's only one agent in the clinic approved to be used as a medicine that targets only one allele, only one mutant, but there are 17 others, and this degrader is giving hope that we can do that. And this is output of our collaboration with Boehringer Ingelheim. This is just to say that the field is now thinking and exploring ideas beyond degradation. This is a review we wrote with a very talented postdoc in the lab, who's now an assistant professor back in the US, where the effector protein could be anything, right? And we could glue for any other types of outcome beyond ubiquitination degradation, and the field is thinking a lot behind that. We can do much better about how we design this molecule. You'll know that these guys got the Nobel Prize just a few weeks ago for the development of new machine learning and new ways to design proteins or to predict how they fold. And now there's a lot of interest in using these methods to understand better how to predict how they bind molecules. And this is his idea that this is just the tip of the iceberg. What I've just told you today, this is just, we're just scratching the surface of how this proximity-inducing agent, molecules that brings protein together, might work and might allow us perhaps to rewire the cell and rewire, how certain processes work, and perhaps lead to gain of function, meaning that you have a modification. In this case, our modification is a compound that we're designing that triggers a process that doesn't happen naturally, right? And so we want to explore that tip of the iceberg, and that's what we're doing at Dundee with our new Center for Targeted Protein Degradation. That's been the innovation I've taken you through. That's the exponential wave of the innovation of the PROTAC and targeted protein degraders. I've told you how we started from an initial, idea, conceptualization, visionary, but not proved for many, many years. In fact, no publications, no real uptake at all, until those years that I mentioned to you, where those pivotal discoveries enabled these initially curious idea, curious minds, but ultimately have to show acid tests that the industry can look at it and say, yes, there could be drugs on it, and then boom, everybody jumps and everybody's on it now. And further innovation I haven't had the time to tell you about, and that's what we're doing now. It's taking the next wave of innovation. That's why we have a center in Dundee, very briefly. It's a pinnacle hub for discovery, training, and translation, led by pioneers in the field with world-leading track record, reimagining collaborative partnerships that deliver. And so we have a lot of partnerships, a lot of interactions with industry. We're based in Dundee. I don't need to tell you where we are, but when I travel around the world, I have to do it. Often, not everybody knows. And that's our new center. We're about 70 people right now. We're very multidisciplinary. We're very proud of the people we've assembled, and the culture that we have, and the drive that we have. I told you about innovate, collaborate, inspire, and I hope you've got a sense about that. The free multidisciplinary of the things that we do, I've given you a flavor of that. And we opened the center in 26 October 2023, exactly a year ago. At the time, George Freeman was MP, and we gave him MZ1. So he held MZ1. And then the week later, we learned that announcement was made that he resigned from the ministry, and then basically made the joke. Essentially, we degraded him as well. And so be careful when you come visit us. We had an amazing symposium in May this year. We brought 300 people in Dundee to celebrate the opening of a center. And this is our partnerships with industry. This is a company I spun out that is now doing very well, growing in Cambridge. We're now building an innovation hub, so the next company stays in Dundee. We get recognition for the work that we do, also the entrepreneurship and the innovation of some of the work that we do. And the collaboration with Berger has been really a trailblazer for that. A bit more about Amphista. These are the snapshots of the partner that we work with. We like to create teams that work in partnerships, that work very dynamically with very ambitious goal to deliver, research-driven milestones. These are some of the examples of those partnerships and some of the numbers that where we deliver in these partnerships, and we want to do more. And we have really an opportunity with this center now to solidify, build on this interaction. But we're not alone. Even just in Europe, we're forging alliance with some other pioneers in this space. Actually, Nico Thoma was the examiner of Charlotte today. And Gerd Winter I have great collaborations with, as well as Ivan Dikic from Frankfurt, and Christina, we have collaboration. So we're very international, we're very collaborative, and I think it's really that outward facing, especially building that European alliance is very important. I'm going to stop here. This is perhaps the most important slide. I want to acknowledge the people without whom none of what I showed you, none of what I told you would have been possible. Their talent, passion, motivation, and drive has made all of the work past and current that I've been able to share some snapshots to you really possible. Former members from my very first ever postdoc, Inge van Molle, she worked for the first time on this. She was my first postdoc, and she saw that key structure of the hydroxyproline. And then lots of people who were former members who now either have their own labs in academia or lead important research, that most of which are still in the field. Current members are collaborators, and I showed you both some academics and industry collaborators, and our funders, without whom none of this would have been possible. And I'm sorry I've taken a few more minutes than expected, but I hope there will still be time for questions. We're happy to take any questions and discuss further. Thank you for your attention.
Thank you. That is cutting edge, I think we'd agree. Yeah. Any questions? Yes.
Yeah. So ubiquitin is literally ubiquitous in nature, from bacteria through higher animals. We know that all proteins have a lifespan within cells. How is the ubiquitination system in proteasome involved in degradation of all sort of degraded proteins within the cell.
Yes.
How would they be targeted?
Can you hear that up the back?
Yeah, it's a great question, but I can also try to not butcher it and try to repeat it somehow. So I think the key question here is, this is an important conserved natural mechanism, right? Is this the only mechanism used by the cell? And how do you deal with it if you want to if you want to degrade a protein that maybe doesn't go by this mechanism? So it's a fantastic, great question. So I didn't say this, I didn't mention this, but it turns out the ubiquitin proteasome system is not the only natural mechanism. There are other mechanisms, for example, autophagy, for example, lysosomal protein degradation. So the cell, for example, goes through these lysosomes, which are organelles, or autophagy as well. Similarly, they engulf bits of the cell, including unwanted protein, into newly formed vesicles, right? So regions where you can separate the interior of that from the exterior, even within the cytoplasm, the cytosol, and then trigger the degradation of that. So, however, it is fair to say that ubiquitin proteasome system is the most prevalent for the cell used for degrading intracellular proteins, cytosolic nuclear proteins. So if you go back to that picture I showed you of the cell, that's typically what how the cell degrades those proteins. And so the nice thing is, even if there is not even if a protein X that sits in the cytoplasm or inside the nucleus, and that's where most processes happen, and that's where most targets are that we want to trigger. Even if a particular protein is not regularly, is not natively going through the ubiquitin proteasome system, we can still hijack it. We can glue it to those ligases, and they find ubiquitin, and it gets recognized, even if it's not naturally. It's amazing. So what we are restricted with this approach, so long as we hijack the ubiquitin proteasome system, is the location of those targets. Because the ubiquitin proteasome system is only intracellular, is not, for example, on membranes, is not in the mitochondria, for example, is not in other organelles such as the endoplasmic reticulum, then we can only target those intracellular proteins that are accessible to it. Membrane proteins, we can target them from inside, but not from outside. But secretory proteins, those are more challenging.
In which case, how are those normal cells normally targeted? Presumably they don't have a target site and a unique ligand to bind.
Say it again. So how normal
So
cells?
how would, if they're being degraded using ubiquitin, presumably do they all have a common site? No. So what's the normal way of ubiquitin actually identifying them? The ubiquitination system identifying them.
Oh, how do we identify proteins that are naturally targeted by? Yeah. That's not easy, actually. And but there's this concept of so-called the degron. So there are regions of the target protein, so-called degron, that get recognized by the system. Sometimes they're in the middle of the protein and they get modified by hydroxy, like the one I showed you. Right? Hydroxyprotein is the degron for that hit. Sometimes they're at the termini of the proteins. Sometimes they get mediated by metabolites, like opsin. Sometimes it's a simple recognition, protein-protein interaction. And what's beautiful about this is we don't need that information in principle.
Yeah.
So our target protein doesn't have to go via that naturally. If it does, it's interesting. And it might be useful to know, but to actually get us to those degraders, we don't need that.
Yeah, so you've got your wee dumbbell, and at one end it's attached to the ubiquitin, and then the other end, okay, it's designed to attach to your target
Yes.
protein.
Yes.
well, how specific can that be? And is it difficult to make that really specific? Because you've got a gazillion proteins in there. And
Oh yeah.
so,
Yeah.
so how do you prevent it gobbling up the good stuff?
Yeah, it's a great question. And so I think the best way to show it to you and to make you think about this is probably, showing you this again. Because, so this is a dumbbell, and we now know it's not a dumbbell, right? And so at one end, so first of all, just to remind everyone, it doesn't bind ubiquitin directly, right? It binds the ubiquitin ligase, it binds the enzyme, right? It binds a protein that then fires ubiquitin, okay? And this is important, the ubiquitin gets built here, and it comes from here. So it comes from this end of that enzyme, okay? So, and then it recruits, you're quite right, at the other end is target protein.
Yeah.
So your question is, I don't just have this target protein, I have hundreds, thousands of other target protein. Haha. So I haven't told you about this issue of target selectivity, but this has been one of the critical discoveries from that structure that we made. Because it turns out that when you have a protein like this, you can already build reasonably good discrimination by already designing a molecule at this end that is pretty selective. Okay? So when I do that, I'm only engaging one or two or three of those proteins, right? But if I have a very selective ligand here, I only engage one. So I'm only engaging one here, then I'm only going to form that ternary complex with that one. Having said that, it turns out that there are ligands here that sometimes we just can't get them to be so selective for a single protein. Why? Because the binding site here is often very conserved, and we cannot discriminate amongst different proteins. And what then happens is in that ternary complex, we make that discrimination. And I didn't have the time to be able to go into the detail, but the way to understand this is actually from that crystal structure that we solved. That's when we first illuminated it. Why? Because the part that binds the ligase is very specific. We made it very specific. It only binds that ligase, so we don't worry about it. But the part that binds this target, this is this BRD4. There's actually two other proteins called BRD2 and BRD3. And so, as you can imagine, they're very similar. And they have the same region, and they have the same binding pocket here. And this ligand for that cannot discriminate those, right? It engages to them equally well. Why? It's explained by this picture. I didn't have the time to go through it. So, you can see here, this is the surface of that protein, and this is where that ligand binds. And these different colors are the conservation of the sequence. So it basically, if it's blue, they're all the same, right? And you can see in the binding pocket, it's almost identical. So the molecule has identical amino acids in your protein to interact with. So it can't discriminate. It's binding to all those targets. And in fact, it's binding 3 of them. What we found was this one degrades more specifically one of them, this BRD4. But we didn't know how and why. But when we saw that structure, that illuminated exactly how and why. Why? Because in the context of the structure, we're now inducing this new interaction with the ligase, with residues that are more in the surrounding of the binding pocket. This is a very difficult concept to get, and it takes time to grasp it, but it's so, when you think about it, it makes complete sense. And now you see those residues, they're shown here in terms of what they interact with in blue, right? And here is the same position, because the protein is drawn in the same way. And you now see that they're green, red, yellow, right? Green again, and yellow again. So they're much less conserved. So they're very different between the different targets. And so what that means is that this protein glues, it forms a more favorable recruitment with that BRD4. That's why it gets degraded preferentially. And so when we basically then extrapolate that, and I told you that we now understand exactly how we do this, and this is the beauty of this approach. We can have the same degrader molecule that engages two different proteins at the binary level, 1-to-1, exactly with the same affinity because the pocket is the same. We can have, for one target, they're flat. They don't do anything. For the other target, fast and potent. That's the selectivity we can get with these degraders. And we get that because of that differential ternary complex and also differential ubiquitination, that firing to the bullseye. Because ubiquitin has to go to a lysine residue, and so whether you have a lysine or not in the right place, you can get more or less ubiquitin. It's fascinating, right?
I can see that. Yeah.
Right up the back, big shape.
So you're using the CRBN and the VHL to attach to these things to fire the ubiquitin. Is there any concern over the lifespan of these? Because they're being used unnaturally, like you say, so they're going to be used extra. So is that going to affect their lifespan in the body?
Oh, it's a fantastic question, right? So you're asking a question about what happens to the natural lifestyle, right, of these ligase now that you're hijacking them to do a different job.
Yes.
Right. So this is going back to the pharmacology and that early slide that I showed you, right, where I was basically showing you, I can't see my mouse, where I was showing you about the, how these work and how this substoichiometric mode of action. So, and remember I told you if you have 100 molecules of your target, you don't need to engage all 100 at the same time for this to work. You may just engage 5 or 10 at any one time because it's catalytic.
Yeah.
Well, you can apply the same principle on the E3 side. Right?
Okay.
So we don't need to saturate all the molecule of our E3 that are inside the cell. At the very low concentration that this drug work, at the very low dose and concentration that they work, they're so potent. And because they're catalytic, all we need to do is to hijack a small portion of the whole population of the ligase. And then we're left with all the rest untouched one that can carry on and do its own natural job.
Yeah, so it's irrelevant.
Huh?
It's irrelevant.
Yeah, it's amazing, isn't it?
Yeah, cool.
And so when you asked me about cerebral and VHL, I thought you were going to ask, are they expressed everywhere? And so are we now degrading the target everywhere?
It was another thought of mine because if you can't, if you've got, you said that it's ubiquitous, so you can't obviously have it active in one cell and not another cell.
Exactly.
So if there's proteins of the similar nature in different cells, then
Yes.
you're going to be attacking those as well.
Absolutely. That's an absolutely key question, really important to be aware of. And it turns out that VHL is expressed everywhere, so cerebellum actually. And so we're now dosing molecules into patients, they distribute everywhere, not just in the cancer cell. So they're going to degrade our targets everywhere, and the answer is yes. So are we worried about toxicity? Yes. So how do we mitigate that? So a number of ways to do that. One is to carefully choose a target protein so that only the cancer cell, for example, gets killed by removing that protein. So only the cancer cell is really vulnerable, right? Is really sensitive and depends on that protein. Whereas normal cells don't really care. That's why we have to be so selective. Because if we start degrading too many proteins, then we might just start killing also other. The other thing is we don't want to degrade adventitiously another protein that we don't want. On the other hand, you're quite right. There are proteins that are important also to normal cells, and if we degrade them, then those normal cells aren't going to be very happy. That's why standard chemotherapy is so drastically toxic, right? Because it's undiscriminatory. And when that happens, then there's really only few approaches that one can think of. One is, for example, try to make it specific for the cancer cell. You can couple your degrader to an antibody. The antibody accumulates in the cancer cell, releases a degrader, and now the degraders only degrades there. That's an approach that is becoming more and more popular now. Or an alternative could be to think about newly free ligases. So this is another thing I thought you might ask. So if VHL and several are only 2 ligases, it turns out in the human genome, we now know there are over 600. So perhaps we can now co-opt with this approach a ligase that is much more restricted, that is only expressed in the disease cell. And then because it's not present in the normal cell, we shouldn't be degrading the target there. That's what also is really exciting about the approach.
I've got a question, if I may. You give the virus example, and talking of hijacking, which they do,
Yeah.
and they've been doing it for 2 billion years,
Yeah.
and are good at it.
Yes.
Is there anything to learn from them in terms of their adhering to these proteins that are trying to kill it?
Absolutely. I mean, absolutely. And there's always more to learn. There's always new opportunities to learn. What viruses typically do, as I mentioned, right, is they co-opt. First of all, with their viral genome, they co-opt the host machinery of transcription translation to then synthesize the viral protein. And then it's those viral proteins that act like a PROTAC.
Right, so they're doing it in a different way.
So they're doing as bioprotacs. So that's another twist, right? It's another angle of basically developing proteins that work like that. They're often called bioprotacs. You can develop an antibody, but antibody is too big. Maybe smaller, a nanobody, so regions of proteins that are very small that can bind your protein, and then you find a way to also bind the ligase, boom, and they act in the middle. So you can learn about that. But then the viral protein might just hijack, a host antiviral protein. That might not necessarily be the target that you want to go after for making an antiviral drug, because they try to do the opposite.
So the approach of these new 40, 50 drugs that you mentioned here are all synthetic.
Are all synthetic or small molecule organic molecules. Yeah. But people are trying to develop these bioproducts, so absolutely.
Yes.
Are these all still in development or clinical trial phase, or have they been approved? Are there specific ones that have been approved?
So, not yet, but it takes a long time to develop drugs, but the outlook is very good. And the field anticipates a potential approval already in 2025. So I just came back from Boston. This was a big targeted protein degradation annual summit event, and so we had a lot of updates from the industry. 2 pioneering companies from the United States, Arvinas, as Great Cruz spin out, and Nurix Therapeutics, shown here, as well as Chimera. These are companies that are developing some of the most advanced agents. Some of these are in phase 3 clinical trials already. It's very exciting. Some of these have shown some toxicity, but no safety concern of toxicity has emerged over the past 5-6 years, because there's something wrong with this new modality, with this new mechanism. So, degrading a protein and generating the proteolytic peptides at the end of that proteasome is not per se a safety concern. It hasn't triggered as a mechanism, as a general mechanism, any concern. So, the toxicity we often see is probably because of the choice of the target, or like any small molecule drug design, at some point something will pop up. And these are small molecules drugs like the conventional drugs. So they face the same issue of managing efficacy versus safety and toxicity. And on the other hand, remember I told you of lenalidomide and how lenalidomide has been approved and has been such a blockbuster drug. So the field has taken that and now expanded on that and created analogs of that and started to find ways to degrade other proteins, not just the protein that cause phocomelia, or the proteins that transcription factor that underpins the mechanism for multiple, to treat multiple myeloma. So it turns out there are now 4 or 5 approved of those smaller lenalidomide-like molecules. Whereas all of these are these sort of 2 heads joined by a linker. That's why I've kept the focus on this, because I think it's just everybody thinks it's just gonna be a matter of when, not of if. And who's gonna be the first approved? There's a bit of a race on that, But once you get that first approval, there's still a lot of skepticism. There's still a lot of, have these molecules, safety, they look too big. There's still skeptics out there, because it's easy to be skeptic until you've shown some success. I think once the first drug will be approved, there will be even more boom. It was a bit like kinase inhibitor. They needed the Gleevec, that first approval, and then 30, 40, 50, 60 approvals followed.
And will the approvals then get faster in the process because it's using the same process and key of the process?
It's a great question. So you're really asking whether there might be the case that, this has been somewhat slow because the bar for safety or the bar for approval has been high because it's a new modality. Not really. I think that there's no doubt, right, that the regulators are looking carefully at this, especially when you've got these things here because of the history of thalidomide, right? So nobody's gonna approve any molecules like this, for example, if they were to use as antimicrobials or pesticides. Nobody, right? Because of the safety concerns, they would not touch that. But, cancer patients that have, been heavily pretreated, maybe 4th, 5th line, they have no other options. I think that's, and now we understand better how to manage the safety concerns of these things. So I think that's really manageable. I think the first approvals are going to allow the companies, for example, that have got that approval to expand to other indications in ways that they can't do until they have that first approval. One of the things I've learned about this is the remarkable modus operandi of how clinical trials work. There's going to be no medical doctors that is going to test a new chemical agent, unproven, unapproved, in clinical trials in any patients until, before they have given them standard of care, right? So some of these agents are being developed for diseases such as, for example, just give an example, the androgen receptor degrader here. And there's a few, and this was the first drug that entered the clinic. The androgen receptor degraders are being tested in patients that have a lot of standard of care. They have first line, they have second line, and in many cases they have already some degraders for these nuclear receptors. And so the bar then is really high. Why? Because every cancer patient develops resistance. And in those first lines of treatments, right, it's easy to develop resistance. And then God knows what happens to the biology of the tumor, right? And you generated a drug that's supposed to work, and you've tested it in cell lines and in animal models, but you haven't really tested on those patients that have been so heavily pretreated. So sometimes the efficacy is very low. But once approved and once shown that they can provide clinical benefit, then you can really eventually expand. One of the really exciting aspects here is with BTK here, is this is a target in several hematological malignancies. And one of the things that patients do, there's some inhibitors that work beautifully. And what those patients do is they develop resistance to those inhibitors. And when they get that resistance, cancer cell, they then relapse. And they then have absolutely no options. These degraders are capable of degrading even the resistant mutants. And then, essentially, that's your only option. And in the field, we're now seeing remarkable, extraordinary responses. So this compound here is a bit developed by this American company. It's actually being first trialed in the UK. And so there's several UK patients in UK hospitals that got this. They started responding back again, and they had no option.
One last question, maybe? At the back, yes.
How long is a protein's average life?
Oh, incredibly good question, a very important question. So you're really asking what's the half-life of the protein, right? The average life of the protein. And this is really important, right? Because we accelerate the death of the protein. Right? We remove it quickly from the cell. But it turns out that many proteins are rapidly synthesized and also rapidly degraded, natively, endogenously. So in a case like that, maybe the average life of the protein could be minutes, right? So a single protein molecule, on average, is made and then degraded only after a few minutes. So a lot of it is made and a lot of it is degraded. On the other hand, there are proteins that are made at some point and they live for many hours, sometimes days. They form very stable complex and the cell doesn't care of them. Let them do their jobs for a long time. So these proteins have much longer half-life. So basically, it very much depends on the types of protein and the types of cells. Some proteins are very short-lived. Some proteins are much more long-lived. Turns out that these degraders work very, very well with very long-lived proteins. Why? Because you have a population of proteins and you now tag them and you find a way to remove them, but then the resynthesis is very low. So you remove them quickly and then they don't come back. It's very durable. Makes sense, right? Short-lived proteins is really hard because the cell makes them really quickly and already degrades them really quickly. And you're trying to tip that balance towards getting rid of it. And the bar to do that is really high. Plus, and we just published a paper called Confounding Factors. When you work on proteins like this, there are lots of artifacts. It's too complicated now to explain. But basically, those proteins that are synthesized very rapidly and degraded very rapidly are much harder to really get rid of. We can still do it.
What a perceptive and incisive question to end on. Thank you very much.
Thank you. Thank you very much.
Alessio.
Yeah, thank you.
Yeah.
Yeah, thank you. Very quickly.
Right. I don't know about you guys, but I kind of felt tonight that I felt very privileged, you know? I felt very privileged to be sitting here with a guy who's at the top of his game, okay? Who's working at the absolute forefront of science, I mean, way beyond my knowledge horizon, and he's working on something that's relevant, you know, that has genuine implications for people like us in, presumably, the not too distant future. So the work that he's doing isn't some airy-fairy, hypothetical, theoretical stuff. This is the real deal that's going to affect people's lives. So anyway, I thought that was quite amazing. We've been organising, me and Claire, we organise these talks. I've been doing it for 10 years now. And each year we try and have a mixture of speakers. We try and have, you know, speakers that are really accessible, popular subjects, and then we stick in some really tough ones, you know. So several of you here will have sat through talks on things like, you know, the quantum physics stuff, yeah, the spooky action at a distance stuff, and some of the really, really difficult topics, you know. This one, I was pretty sure when Claire came along and I did my research, and I looked and I thought, well, jings, you know, that's going to be a tough one, challenging. But you know, when you've got a speaker who's a good explainer, you're able to go right to the forefront of science to really, really complicated stuff, and it makes sense, you know. So for me anyway, I know a lot more about this subject than I did an hour or so ago, and I'm not sure what else to say other than I thought that was just really good. So please, round of applause for our speaker. Thank you very much.